Today we'll be talking about muscle cell depolarization. So in case of muscle cell depolarization, what really happens is it all starts from the spinal cord. The spinal cord, the ventral portion of the spinal cord has alpha motor neurons. These alpha motor neurons carry signal to the myofibrils. So it can carry signals to many myofibrils, that is furthest and the nearest also, anywhere. So whole myofibril bunch together is known as a motor unit. Then these signals are then passed on. If you look at the microscopic version of this myofibril, you can see an axon and a neuromuscular junction. So usually we call it as presynaptic and a postsynaptic area, but instead of this postsynaptic area, since it is a neuromuscular junction, we call the postsynaptic space has the motor end plate. So you can see an axon here. This axon will carry the action potential. When we look at it at a microscopic level, the axon will carry the action potential, which further will open the voltage gated calcium channel and allows, so this is depolarizing in and this is getting open. The calcium channels are getting open and it causes a calcium induced acetylcholine vesicle exocytosis. That means as the calcium is increased inside of the axon, it causes the vesicles of the acetylcholine to exocytose and then reach the synaptic cleft. So as we can see, we have reached the synaptic cleft, and these acetylcholine will bind to certain acetylcholine receptors which are present on the motor end plate. So this is known as ligand gated sodium channels. These things are known as ligand gated sodium channels because as this acetylcholine is acting as a ligand, which then gets binded to this channel, which will cause sodium release and the action potential will be carried on. So as you can see, this motor end plate will also have a T-tubule. Now we'll be talking about this T-tubule. So as the action potential reaches the T-tubule, it stimulates the DHPR receptors, dihydropyridine receptors gets depolarized, which causes, so you see this depolarization of dihydropyridine receptors with the muscle cells that is present on the T-tubule. These T-tubule DHPR receptors are coupled with something known as RYR, which is ryanodine receptors, which are present on the sarcoplasmic reticulum. So if we take this as a muscle cell, it has got sarcoplasmic reticulum. This is the sarcoplasm. This is the sarcolemma, the layer, the border. Okay. So yeah. So this these two receptors are coupled. So whenever this is depolarized, DHPR is depolarized, it also depolarizes ryanodine receptors, which ultimately opens. So when this is opened, the calcium inside, the sarcoplasmic reticulum is filled with calcium, and the calcium inside the sarcoplasmic reticulum will come outside into the sarcoplasm. So that is what is happening. There is increase in calcium into the sarcoplasm. This is known as muscle cell depolarization, which further causes muscle contraction, muscle relaxation, and stuff like that. So we need to know the basics of this muscle cell depolarization for anything else to for us to understand anything else.